Let's discuss the nomenclature of the genetic abnormalities found in alpha-thalassemia genes. So here's how you may find the nomenclature of the different genes located in the alpha-globin. As you see here, usually what you tend to see is two different alpha genes on one side and then two other genes on the other side. These are two of the genes that are located in one of our chromosome 16, and then this is the other chromosome 16. And that means that we have a total of 4 different genes of alpha-globin in our body. Now, let me give you a first example. We have something called a silent carrier. And the silent carrier has only one of the 4 genes affected by alpha-thalassemia. So, for example, you may have a description something like this, will be A minus AA. And what this means is that you have one affected gene while the other 3 are normal. There are different combinations that you may see. For example, you may have a minus A, AA. You may have an AA minus A, or you may see an AA, A minus. If you see here, we are just basically combining the different possibilities of which of those genes may be affected. When we have only one gene affected, usually this tends to be silent, as we mentioned before. It barely causes any consequences, and the main thing that you may see is a low MCV when you check CBC. The second status that you may find will be alpha-thalassemia trait, also called alpha-thalassemia minor. And here what you have will be 2 gene mutations affecting the alpha-thalassemia globins. So here you have an example in which you may have something like this, A minus, A minus. Now there is a difference between having one abnormality in one side of the line and 2 abnormalities on the other side of the line. And this is mostly important in regards to the name that we will have. For example, if you have something like A minus, you have one abnormality on one side and one abnormality on the other side. And when that occurs, you will call that a trans abnormality. The difference between that and, let's say, for example, a cis abnormality will be that you will have both of the alpha-globin genes affected in the same side of the line, as we see here. And this is important mostly for different mutational combinations that we may see. Other possibilities that we can have here will be minus A, minus A. We may also see a cis combination like this, the other possibility of cis combination, and so on. Now, the third possibility here will be to have 3 out of the 4 alpha-globin genes affected. In this case, we will call that hemoglobin H, and this definitely has more clinical consequences that we will discuss later on. So here you may, for example, see something like this, A minus, minus, minus. You may see a minus A, minus, minus, and so on. And finally, we have last, which is the alpha-thalassemia major, or also called hemoglobin BARTs. In this case, what we tend to have is, unfortunately, 4 genes that are affected by the condition. This is severe, and this is what will cause hydrops fetalis. I hope this helps you understand a little bit more of the nomenclature of these different genetic mutations that we may see in alpha-thalassemias.